Okay, good afternoon and welcome to the session. I hope you have a wonderful day so far. So yeah, the kangaroo leech is a very interesting creature. So in English we have this saying that says, curiosity killed the cat, and we as researchers just ignore that and try to make a living of it. Yeah. So, and my curiosity led me to this project. This is just an outline of what I've done. I'm an honours student now, so this was based on my honours project. Yeah, so see just the overview, my aims and objectives, and then the conclusion. So this is the kangaroo leech, Marsupioptala africana. As you can see, it has an inferior, poorly defined anterior sucker, which it uses to feed on the blood of invertebrates such as amphibians and fish. So this is just a short life cycle. As you can see, the leech feeds on amphibians, and it has a very interesting phoretic association with the freshwater crab, Poteminotus parulatus. Also just note that after the leech detaches from the frog, copulation takes place. That would be important further in the presentation. So this is why it's called the kangaroo leech. It broods its young in a pouch in the abdomen, and just when the young are fully grown and developed, they explode out of the pouch onto the amphibian host. So this is the association of the leeches with the frog. As you can see, they feed on the frog, and he doesn't look very worried, but he indeed is. Okay, so this is another interesting thing about these leeches are they have a hypodermic implantation of a spermatophore, which means that the spermatophore is planted on the dermal wall of the leech, and then the sperms are released into the body, and the sperms find their way to the ova and then fertilize it. I'm just going to show a short video. They warned me to just say parental guidance. I hope the video works. Doesn't. Just go on. The video is in fact of two leeches copulating, so I hope it works. Okay, doesn't seem to work. Okay. But what happens is that bodies of the leeches entwine, and the spermatophore is planted on the body of the other leech, and yeah, that was what the video was about, not that romantic. But so as you can see, multiple spermatophores can be implanted on the leech. So there's not a specific place where they are implanted. And this is just another example. Just to note, these leeches are hermaphroditic, so their sexual reproductive systems are very complex and elaborate. So when the sperms find the ova, they are fertilized, and the fertilized eggs are transferred into the brood pouch, which you can see over there, through the female genital opening. It's a complex system, so I'm not going to go into detail. And this is a brooding leech. As you can see, the brood pouch is no true opening. So the young are in there developing now. At this stage, a life stage specific phoretic association is possible to occur between the leech and the crab, which basically means that the leech uses the crab as a taxi to get to aerated water, so the young can develop. So this is just examples of the leech on the crab. So the crab goes to water where there's oxygen. The leeches need the oxygen to incubate the young, for the young to grow. These, the family of leeches are ecologically and economically important, as they are widely distributed in almost all freshwater habitats, and they are stress indicators of those habitats. And because they feed off the blood of invertebrates, they can also act as a vector or host for blood parasites. And yeah, identification and classification have been problematic because not a lot of studies have been done on this specific leech. So I have 4 aims and objectives I looked at in my specific study. These are the 4. We are going to refer to them later on as well. So for my study, the distribution of this leech is not known. We don't know much about the distribution, but what we do know, luckily for us, there are two populations in the Northwest University Botanical Garden, and those were the two sites we chose for the study. So what we did was we decided to catch the phoretic host, which is the crab, and we used baited bucket traps as well as dip nets to catch the crabs. This was done once a month for this entire year. So these are my first two aims. The morphology, to determine the morphology and the structure of the spermatophore, as well as to determine the nutrient providing strategy of the brood pouch. So how does this mother provide nutrients to the young in the brood pouch? When we collected the crabs, we checked the crabs for where the leeches occur. So we looked on each segment and saw, okay, there's 2 or 3 leeches on this segment, and we noted it. And we collected the leeches with forceps. So we prepared the leeches by directly after collection, we looked at them under a microscope and then checked which have spermatophores, and we fixed those for histology and microscopy work. So for histology, this is just an outline of what we've done. I know some people are better experts in this than I am. So we fixed them in boron solution, we dehydrated them in alcohol, cleaned them with SAEON, embedded them in paraffin wax, and then we made thousands of cuts, and the end result was this microscope plate where we just examined the leech. For microscopy, we fixed them in TOT solution, we had dehydrated them in alcohol, we critically dried them, then we inspected them with a microscope containing a camera, took some photos, we then coated them in gold. I wanted to coat my ring, but I weren't allowed, and then we did some scanning electron microscopy. So what we found in this specific, I can say experiment we've done, is after we detached the leeches from the phoretic host, copulation did occur. So it doesn't just happen of the infective host, which is the crab, which is the amphibian. So they also copulate after they detach from the phoretic host, which means that this is not life stage specific attachment. So they don't only attach to the crab for incubation periods. And possible explanations can be because premature detachment occurs, so they did attach to the amphibian host, but they fell off and then didn't copulate, or they didn't reach sexual maturity, or there was a lack of amphibian host, or multiple matings can occur. None of these have been verified. So this is just the implanted spermatophore. As you can see, there is 3 distinct regions. We will go into more detail. The first region is the germinal region, which is that region which consists of 2 oval bundles of sperm and is separated by a cleft. And this is just a cross-section through the germinal region of the spermatophore. And as you can see, there's aggregations of sperm inside the spermatophore, which is released into the body of the leech. This is just the proximal attachment region, which has these hairy, sticky structures, which keeps the spermatophore attached to the body of the leech for several hours. This is a cross-section through the proximal attachment region. As you can see, it has a subtriangular rim, and this is a funnel, funnel-like invagination formed by the spermatophore, which is going into the body. Okay, this is a cross-section through the body of the leech, and on the top there, that's the proximal attachment region, and there's the aggregation of spermatozoa. So these sperm go through the body and they find the ovary and fertilize it. This is just a close-up of the aggregation. And this is the last region of the spermatophore, the distal horns region. And this is only presumed to form when the one leech attaches the spermatophore to the other leech. This is a sagittal region, a section through the adult. This is the brood pouch, and in here there are more than 5 babies. There you can see that the baby is attached to the adult by the posterior sucker. This can indicate that the adult feeds the young through the posterior sucker, or provides it with nutrients. So my second aim and third aim was to predict the preferred site on the verticoast and to see if this phoretic association is life stage specific. What I've done here, I've made a behavioral experiment. This was the setup, so we had 5 crabs in there and 5 individual leeches that we've placed on the crabs. So remember I told you that when we collected the crabs, we checked where the leeches occurred on the crab. So then we placed the leeches, the individual leeches, on the sites where they were least found. So the less preferred sites. So these were the sites, those 2 sites. So we monitored them and checked where the leeches were every 10 minutes. And this is what we found. 80% of the leeches located their preferred sites on the crab in under 30 minutes, which is like a new world record or something. And this could be because of predation avoidance. Maybe there's better oxygen. Studies have to be still done on this. So the 3 mostly preferred sites was the postlateral margin of the carapace, the mirrors, and the bases. So if I was a leech, these would be the sites I would prefer to. It's out of reach of predators and even the crab itself. And 50% preferred the postlateral margin of the carapace. So in conclusion, the spermatophore is a 3-region structure. It consists of the germinal region we saw, the proximal attachment region, and the distal horns region. The adult provides nutrients to the juveniles through the posterior suckers, and the phoretic association is not life stage specific. And the leeches have 3 preferred sites which they migrate to. And research is not easy, but with appropriate training, experience, and mentorship, one can profoundly change the world. So at this point, I would just like to thank these people. And thank you for listening. And also remember to like our Facebook page. Thank you.